Hey guys, Mr. P. In this video, we're going to talk through the entire aerobic cell respiration pathway from start to finish. We're going to do it in a simple drawing in order to identify the relationship between all of the individual processes. So, the aerobic cell respiration pathway begins with glycolysis. Glycolysis happens in the cytoplasm, which is the fluid that is inside the cell membrane but surrounds all of the organelles, including the mitochondria. Prep step, which is also called the link reaction. It occurs in the matrix right before the Krebs cycle. Krebs cycle will follow. That again occurs in the matrix. And then we have the ETC/chemiosmosis, which occurs in the cristae membrane. So these are our steps. Step 1, glycolysis. Step 2, prep step. Step 3, Krebs. Step 4, chemiosmosis. Now the first thing we need to do is obviously draw glycolysis. Now there is a previous video that goes through the intricacies of glycolysis. If you need more help, check out that video. But glycolysis is a process where a 6-carbon compound called glucose is going to be oxidized, meaning we're going to take electrons from it and we're going to ultimately turn it into pyruvate. Now we do that through a series of intermediate steps. And the first of those steps is we have to take glucose and add phosphate to it. And so we add phosphate to it from ATP. So we will bring in 2 molecules of ATP. Those will convert into 2 molecules of ADP. They will add the phosphates from each of the ATPs to the ends of this molecule, and that will leave us with a 6-carbon compound with 2 phosphates added. That molecule is called fructose 1,6-bisphosphate, and that molecule will immediately split or lyse, and we will be left with 2 halves that each have 3 carbons and 1 phosphate each. We call those G3P, or glyceraldehyde 3-phosphate. Now again, like we did in the other video, this marks the end of our energy investment phase, which is we have to add 2 ATPs or spin 2 ATPs so that we can ultimately get energy from the molecules later. Once we have our G3P produced, we're going to oxidize it, meaning we're going to bring in our oxidizer. We will bring in a molecule of NAD+. That molecule will grab a pair of electrons and a hydrogen ion from G3P, convert itself to NADH. That is a full taxi. Again, we have videos on that. And that molecule is off to the electron transport chain. We will come back to that in a minute. Again, it occurs on this side as well. NAD+ comes in. It is converted to NADH. That NADH is off to the ETC as well. It grabs its pair of electrons and a hydrogen ion and therefore takes those electrons and hydrogens from those G3P. So from each G3P, we have produced 1 molecule of NADH per G3P. And because we take those electrons from G3P and we take that hydrogen from G3P, it allows a phosphate, which is freely floating and freely available in the cytoplasm, to join these G3Ps, and we end up with a molecule that has 2 phosphates and the 3 carbons it had to start with. So our molecule is completely saturated with phosphate, and now we need to pull those off, and we're going to produce 2 molecules of pyruvate, which notice is a 3-carbon compound. It does not have any phosphates, and that's because we are going to bring in 2 molecules of ADP. We're going to pull those phosphates off, add them to the 2 molecules of ADP, and we end with 2 molecules of ATP, high energy compound. Again, same thing over here. We're going to pull these phosphates off. We're going to add it to 2 molecules of ADP. Those are going to be converted into 2 molecules of ATP, and we are left with our 2 molecules that are 3 carbons each called pyruvate. That is the end of glycolysis. we call this, underneath this dotted line, the energy payoff phase. We initially invested 2 ATPs up here. We're going to gain a 4 ATP gross profit. But because we invested the 2 ATP up top, we're going to subtract those from our gross, and we are going to get our net profit, which is a positive 2 ATPs. Now, this entire process happens in the cytoplasm, which is the fluid outside of the organelle, specifically mitochondria. And so the whole purpose of this is to take glucose and convert it into 2 molecules of pyruvate. One of the benefits of doing this process is we are going to produce a little bit of ATP. As we do that, we will talk about fermentation in a later video, which is what happens after this step if oxygen isn't available. But again, we talked about at the beginning of this video, this being the aerobic pathway. And so now, once we have pyruvate produced, we're going to go into the second step, which is the prep step. And once our pyruvate is made, it's going to drop right into a process called the prep step, in which the pyruvate that was made during glycolysis, again times 2 because we made 2, we're just going to draw it once, is going to be oxidatively decarboxylized. Again, we're going to pull our oxidizer, which is NAD+. NAD+ is going to come in and be converted into NADH. Again, just like these other ones, it's going to be off to the ETC. It's going to grab its pair of electrons and a hydrogen ion. And because it is oxidatively decarboxylized, we're going to cut a carbon off in the form of CO2, which is going to be released to the atmosphere, and it's going to turn that 3-carbon pyruvate into a 2-carbon acetyl, which is then going to be added to by bringing in coenzyme A and producing a molecule called acetyl-CoA. That marks the end of the prep step. Again, it happens twice because we produce 2 pyruvates. Now we have 2 acetyl-CoAs, and those acetyl-CoAs can enter the Krebs. Once the acetyl-CoA is produced, it's going to enter the Krebs, and waiting for it in the Krebs is a 4-carbon molecule called oxaloacetic acid or oxaloacetate. That 4-carbon compound is going to be added to with the 2-carbon acetyl. The coenzyme A is going to leave and return back to where it is needed within the prep step. The acetyl is the 2-carbon, oxaloacetic is 4. That's going to produce a 6-carbon citric acid. And once the citric acid is produced, it's going to be oxidatively decarboxylized, which means we're going to bring in our oxidizer. NAD+ is going to be producing NADH. NADH is going to be off to the ETC, just like all the other ones. It's going to grab a pair of electrons and a hydrogen ion, again, just like the other one. It's going to decarboxylize or kick off a CO2. That CO2, again, is going to be released to the atmosphere. Once we oxidatively decarboxylize our citric acid, we're going to be left with a 5-carbon compound because we cut one of the carbons off. And if we started with 6 and cut one off, we now have 5. That is called alpha-ketogluteric acid or alpha-ketoglutarate. It's just simply put AKA. Once we have our AKA produced, we're going to oxidatively decarboxylize it again, which means we bring in our oxidizer, NAD+. That is going to be converted into NADH. Again, off to the ETC. It's going to grab a pair of electrons and a hydrogen ion. Again, it's also going to cut a carbon off because this is a decarboxylation event. And if we cut one of the 5 carbons off, we're going to be left with a 4-carbon compound called succinic acid. Now, it is important to note that at this step, there is also an extra ATP produced. And so 1 molecule of ADP will be converted into 1 molecule of ATP. That is due to SLP or substrate level phosphorylation. It happens as a way to produce bonus ATP. Once our succinic acid is made, we're then going to just oxidize it at this point, meaning we're not going to decarboxylize it. We started with a 4-carbon oxaloacetate. We are now at a 4-carbon succinic acid, so we don't want to kick any more of those carbons off. We're going to end with a 4-carbon malic acid. But in order to get succinic to malic acid, we're going to oxidize it. And so at this stage, we have a different oxidizer called FAD. That is going to be converted into FADH2. This oxidizer is going to grab a pair of electrons and 2 hydrogen ions. Notice FAD to FADH2, we added 2 hydrogens instead of just adding 1. So 2 electrons and 2 hydrogen ions are going to be added to FAD, converting it into FADH2. That will turn our succinic acid into malic acid. Once we have our malic acid produced, in order to get back to our oxaloacetate, we are going to oxidize it one more time. In this case, we're going to bring our familiar oxidizer, NAD+. That is going to be converted to NADH. It's going to grab a pair of electrons and a hydrogen ion and will therefore turn malic acid back into oxaloacetate. This is the Krebs. This entire cycle happens in the matrix. The whole purpose of this cycle is to produce these high energy electron carriers. This one is off to the ETC, this one is off to the ETC, and this one is off to the ETC. Once we produce all of these high energy electron carriers, all of them will be flooding the electron transport chain, which is located in the cristae membrane. And so if we go up here into this kind of void and we draw our membrane with our ATP synthase enzyme, which is a fancy enzyme embedded in this membrane, it does have a tunnel in which ions can flow through. This is called ATP synthase. There is an electron transport chain embedded in the membrane next to an ATP synthase. This would be our inner membrane space, and this would be the space between the outer and inner membrane. This would also be the space in which the high H+ ion concentration would be maintained. And so every single one of these NADH molecules is going to come up here. It's going to drop off a pair of electrons and a hydrogen ion. It's then going to be sent back and it's going to be converted back into NAD+. Now these electrons are going to jump onto this electron transport chain. The hydrogen ions are going to be pumped back to the high side, utilizing the energy lost at each step by those electrons. Once these H+ ions are pumped back to this high side, they're going to accumulate in this inner membrane space. Again, this is the high concentration. The electrons are going to be picked up by our final electron acceptor, which is O2, oxygen, which is why we as aerobic organisms breathe in O2. Once the electrons are picked up by the oxygen and 2 H+ ions are picked up as they flow through ATP synthase, O2 plus the electrons and 2 hydrogen ions is going to be converted into an H2O molecule, which is water, which is going to be released every time we exhale with the CO2, which is one of the reasons why we produce water vapor in our exhalant when we exhale. These H+ ions are going to flow through this ATP synthase on their way from high to low concentration. That's down their natural concentration gradient, high to low. They're going to power the phosphorylation of ADP to ATP utilizing the enzyme ATP synthase. This is an enzyme that phosphorylates ADP and inorganic phosphate and turns it into ATP. This process is constantly flooding our mitochondria and therefore our tissue cells with usable energy in the form of ATP. Once those hydrogen ions are passing through ATP synthase, producing ATP, they're either going to be picked up by our oxygen or they're going to be pumped back to our high side in order to be maintaining that concentration and therefore maintain ATP production. This is the entire pathway from glucose through pyruvate, producing a little bit of energy, down through the prep step where we take our pyruvates, turn them into acetyl-CoAs, take our acetyl-CoA, put it into the krebs, produce a whole bunch of NADH and FADH2, send those FADH2 and NADH molecules to the ETC in order to produce a bunch of usable energy in the form of ATP. That's it for this video. If you learned something, give it a thumbs up. Leave any of your questions in the comments. Consider subscribing to the channel. We'll see you later.